Imagine a patient is rushed into the emergency room. They're clutching their chest, struggling to breathe.
Right, an absolutely critical situation.
Yeah, and in that moment, diagnosing a myocardial infarction doesn't just depend on, an EKG or a stethoscope. It actually depends on detecting a tiny, completely invisible enzyme that's leaking from their dying heart muscle.
Leaking directly into their bloodstream, exactly.
Right. So if you are a medical student prepping for your boards or a young professional stepping into the clinical lab, or even just a dedicated self-taught learner trying to figure out how modern medicine works at the molecular level, well, you are in the right place. Welcome to the Deep Dive.
Glad to be here.
Today we are taking on the 7th edition of Principles and Techniques of Biochemistry and Molecular Biology, edited by Keith Wilson and John Walker.
Which is, it's essentially considered a foundational text for anyone in the medical sciences.
It's definitely.
Because modern clinical practice is, well, it's entirely dependent on systems biology and proteomics. You really cannot grasp a patient's pathology without understanding the molecular landscape.
And understanding how we actually measure that landscape, right?
Exactly. Every lab result on a patient's chart comes from these very specific physical techniques.
Okay, let's unpack this. We're going to trace the entire journey of modern medicine today. We'll start at the absolute bedrock, how we mathematically validate lab data and then move through the physical mechanics of isolating and manipulating cellular machinery.
Right, the benchtop stuff.
Yeah, the benchtop stuff, before finally arriving at the clinical applications, like how we find specific bottlenecks in a disease pathway to invent a new drug.
And that progression from the bench to the bedside is so crucial. Before a clinician can even trust a blood test or, say, prescribe a novel therapeutic, that underlying biochemical data must be pristine.
Sustained, yeah.
Because if you're working with biological molecules, you're dealing with weak electrolytes. Their ionic status, their physical shape, their function, all of it will fundamentally change if the pH of their environment shifts even a little bit.
Which means before you can even run an experiment, you need a buffer.
Absolutely.
It's not just about mixing chemicals. It's about stabilizing the pH so your proteins don't completely denature and fall apart. And the bedrock math governing this is the Henderson-Hasselbalch equation.
That classic.
Yeah, it defines the exact relationship between the pH, the acid dissociation constant, and the ratio of the conjugate base to the weak acid.
Right, and understanding that equation allows you to engineer an environment that resists pH changes, even when you introduce other variables.
But even with a perfect buffer, any quantitative lab measurement carries some inherent uncertainty, right?
Oh, definitely. The text really emphasizes that we have to mathematically define what the true value is amidst unavoidable experimental error. Like in biology, if you repeat a measurement, the variance typically follows a Gaussian distribution.
A normal bell curve.
Exactly. The data clusters around a mean.
Right, but what I find so fascinating is how we apply this clinically using confidence intervals. So, for example, if you draw a fasting serum glucose sample, you aren't just getting a single absolute number.
No, you're getting a probability.
Yeah. By calculating the standard deviation, you define this highly probable target area. You might determine there's, say, a 95% chance the patient's true glucose concentration falls between 2.22 and 2.62 millimolar.
And defining that target area is what allows us to compare different analytical methods.
How so?
Well, say a lab wants to upgrade from the older glucose oxidase method to the newer hexokinase method. They run a student's t-test.
Oh, right.
Yeah, this compares the experimental means of the two methods to prove they yield statistically identical results, at least at a specific confidence level.
But what if one of those results is just, wildly off? You don't use a t-test for a massive anomaly, right?
No, for that you use a q-test.
Right. It's a null hypothesis test designed specifically for a suspect value.
Exactly. You mathematically determine if that weird outlier is just an extreme natural variance or an actual experimental error that you can safely throw out.
But to even trust those data points in the first place, your machines have to be perfectly calibrated, which involves least mean squares linear regression, or LMSLR.
Yes. And this raises an important question. How can we trust clinical data if the underlying method hasn't been rigorously validated against external quality assessment samples?
Right. And I actually have to pause on LMSLR, because let's be real, why does a clinical practitioner need to know LMSLR? The math sounds incredibly abstract.
It can, yeah.
But the way I like to think about it is it's like trying to draw the fairest possible straight line through a scatter plot of bullet holes on a paper target.
Okay, I like that analogy.
Right. The shooter was aiming for the center, But the wind, which represents our unavoidable experimental error, blew the bullets slightly off course. So LMSLR basically calculates the exact line that minimizes the distance between all those scattered holes. It mathematically accommodates for the wind.
That is a highly accurate way to visualize it. And clinicians really rely on that line because if that calibration curve is drawn incorrectly, the patient's diagnostic results will be fundamentally wrong.
Wow. Yeah.
And, time-related changes or sample loss can warp that curve during a run, which is why we use internal standards. We often mix structural isomers of the target analyte directly into the sample itself.
Oh, because the isomer behaves identically to the analyte.
Exactly. So any loss in the standard tells us exactly how much of the target molecule we also lost, which allows us to correct the final calculation.
That's brilliant. Okay, so we've secured the math. We know how to validate the data and hit the same true value as the external quality assessment labs. But how do we physically get our hands on the cellular components we want to measure?
Well, the primary physical technique is centrifugation.
Spinning things really fast.
Very fast. We use preparative centrifugation to physically fractionate something like, say, skeletal muscle tissue. We spin it to pull different organelles or proteins down into a pellet based on their sedimentation rates.
Okay, but I want to push back on the other method the book details, which is analytical ultracentrifugation.
Okay.
Because it uses absolutely microscopic sample sizes, like maybe 20 to 120 cubic millimeters.
Tiny, yeah.
So if the sample is that tiny, you clearly aren't trying to separate a bulk protein to use in another experiment. What is the point of spinning something if you aren't separating it?
Because you aren't spinning it to separate it. You are spinning it to watch how it behaves under immense gravitational force in real time.
Wait, watch it while it spins.
Yeah. Analytical ultracentrifuges have built-in optical systems, so by monitoring how a macromolecule moves through the fluid while it's spinning, we can determine its relative molecular mass and its physical shape in its native, completely unaltered state.
That is wild. And once we know its mass, we usually want to see it, right?
Naturally.
So we move past basic light microscopes into optical sectioning, like confocal microscopy, that uses lasers to block out out-of-focus light so we can watch cellular dynamics happen inside living cells.
Right. But to see the true ultrastructure of a cell, like the really fine details of its organelles, we have to transition to the electron microscope.
And here's where it gets really interesting, because electron microscopes require operating in a high vacuum. And historically, you had to treat the tissue with these super harsh chemical fixatives just so it would survive the beam.
Yes, which caused a lot of problems.
Right. How do we know we're looking at real biological structures and not just artifacts, like fake structures completely fabricated by the chemical prep process itself?
Well, avoiding those artifacts is exactly the driving force behind modern electron tomography. To solve the chemical fixation problem, researchers developed high-pressure freezing.
Okay, how does that work?
Instead of soaking the tissue in chemicals, the specimen is subjected to immense pressure and frozen in literally milliseconds.
Milliseconds, wow.
Yeah. The water molecules freeze so rapidly they don't even have time to form ice crystals, which would normally rupture the cellular membranes. Instead, the water turns into an amorphous glass.
Oh, so no ice shards tearing the cell apart.
Exactly. It preserves the biochemical state of the cell exactly as it was in life, completely eliminating those chemical artifacts.
That is incredible. So we isolate the cells, we freeze them without destroying them, and now we move to manipulating their blueprint. We're diving into DNA, RNA, and protein engineering.
And the basis of all genetic engineering really relies on the degeneracy of the genetic code.
Right, the redundancy.
Yeah. DNA has 4 bases, creating 64 possible triplet combinations, or codons, yet life only uses 20 amino acids. So that means multiple different codons can translate into the exact same amino acid.
And we exploit that degeneracy to design synthetic probes that can hunt down specific genes, even if there are, slight sequence variations.
Precisely.
But to study those genes, we need billions of copies, which brings us to PCR, the polymerase chain reaction.
The workhorse of the lab.
Absolutely. And it's entirely driven by thermal cycling. You heat the sample to denature or melt the DNA into single strands. Then you cool it down so synthetic primers can anneal or bind to your target sequence. And finally, you warm it up slightly so an enzyme can extend the new strand. You repeat that 30 times, and boom, you've amplified the gene.
And once it's amplified, we use cloning vectors to insert that foreign gene into a host cell, like a bacteria, to make it produce a specific protein.
But you don't want the
Okay.
bacteria producing it constantly,
Oh,
right? That would waste energy.
right. So we use specific regulatory sequences like the lac operon from E. coli. It basically acts as a biological switch.
Oh, I see.
We can grow a massive vat of bacteria, and when we are perfectly ready, we introduce a specific chemical that flips the lac operon switch, forcing the bacteria to suddenly express the foreign gene all at once.
That is so smart. And if we want to test how a protein works, we might actually want to intentionally break it by causing a specific mutation.
Yes, site-directed mutagenesis.
Yeah, exactly. The book highlights overlap extension PCR for this. You basically run two separate PCR reactions to create two fragments of a gene that overlap in the middle, but you intentionally synthesize your primers to contain a mismatched base pair, a mutation right in that overlapping region.
Right.
So when you fuse the two fragments together, you've created a custom site-directed mutation.
But having a mutated gene sitting on your bench doesn't help if you can't get it inside a living cell.
True.
We use several delivery mechanisms, but the ballistic technique, the gene gun, is structurally just fascinating.
It is literally a microscopic shotgun. Like you take microscopically small titanium or gold pellets, you coat them in your engineered DNA, and you just fire them into target tissues at high velocity using a pressurized burst.
That's exactly what it is.
But my immediate thought reading this was, wouldn't that just completely shred the cell into pieces?
You would think so, but the physics are actually so precise that the microprojectiles pass right through the cell membrane, and the lipid bilayer simply reseals itself behind them.
Mind-blowing.
It really is. And if we connect this to the bigger picture, these fundamental cloning and delivery techniques are exactly how we create transgenic animal models to study human pathology.
Oh, right, like inserting human genes into mice.
Exactly. For instance, to study the mechanics of human hypertension, we can engineer a transgenic mouse that expresses the rat angiotensinogen gene. Or, for atherosclerosis, we express the cholesterol ester transfer protein, the CET protein.
Giving us a living system to test cardiovascular drugs.
Exactly.
But it's not always about adding a gene, is it? Sometimes it's about turning one off, like with RNA interference or RNAi. It's a brilliant mechanism.
Very powerful.
We introduce a synthetic piece of RNA that perfectly matches a specific messenger RNA in the cell. When they bind together, it creates double-stranded RNA.
Which the cell does not like.
Not at all. The cell's natural defense system recognizes double-stranded RNA as a viral threat and just chops it up. It effectively destroys the blueprint before the protein can even be built.
And silencing the blueprint is powerful, but to fully comprehend the disease, we really have to analyze the physical structures themselves, the proteins.
Right, decoding the proteome. And the heavy hitter here is mass spectrometry, specifically techniques like MALDITO.
A mouthful.
Yeah, it sounds super intimidating, but the mechanics are brilliant. It stands for Matrix-Assisted Laser Desorption Ionization Time of Flight.
Right.
You basically embed your protein sample into a chemical matrix and hit it with a laser. The matrix absorbs the laser's energy and vaporizes, which gently carries the intact protein molecules up into a vacuum tube.
Without destroying them.
Exactly. And then you just measure their time of flight. The lighter the molecule, the faster it flies down the tube to the detector, which gives you an incredibly precise mass sequence.
And mass spec gives us the primary amino acid sequence, but, a protein's function is dictated by its three-dimensional shape.
Right, how it folds.
Yeah. To determine its secondary structure, we use circular dichroism, or CD spectroscopy. Proteins are chiral molecules, meaning they are asymmetrical.
Like a left and right hand.
Exactly. Because of this, they absorb left circularly polarized light differently than right circularly polarized light. So by measuring that difference, we can see if a protein is folded into alpha helices or beta sheets.
And for the absolute atomic structure, we use X-ray crystallography. You crystallize the protein and literally shoot X-rays at it.
Which bounce off the atoms.
Right. The X-rays bounce off the repeating atomic planes in the crystal, creating a diffraction pattern. The underlying physics here is Bragg's law, which calculates how those X-rays undergo constructive interference.
And computers map it.
Yeah, by reading the angles of that interference, computers can map the exact 3D coordinate of every single atom in the protein.
Understanding how physical energy interacts with biological tissue is also the foundation of macro-scale clinical imaging, like magnetic resonance imaging, MRI, which is highly detailed in the text.
Yeah.
It functions by detecting the 1H resonance of water molecules.
Which blew my mind, because water makes up, 55 to 90% of our soft tissue, depending on the organ?
That's right.
So mapping the specific resonance of those water molecules under a massive magnetic field essentially creates a flawless map of the body's internal anatomy.
It's just a really elegant application of molecular physics. The text also details the use of radioisotopes to track biological molecules through a metabolic pathway.
Like glowing breadcrumbs.
Essentially, yeah. But to use them safely and accurately, you must understand a given isotope's half-life. Phosphorus-32, for example, has a half-life of exactly 14.2 days. We measure its decay in disintegrations per minute.
But there's a physical limitation there called quenching, right?
Yeah.
Like if you
Yeah.
put a biological sample into a detector, the sample's own color or physical density can literally block the light emitted by the radiation,
Yes.
causing the machine to undercount the radioactivity.
Quenching definitely distorts the data. To solve this, clinical labs use scintillation proximity assays, or SPA.
How do those work?
Well, we use microscopic beads containing a scintillant that emits light only when a radio-labeled molecule binds directly to its surface.
Oh,
Yeah. Because the light is generated at the exact site of binding, background interference is virtually eliminated. It allows for mathematically precise, high-throughput drug screening.
which leads us perfectly into systems biology.
Hmm.
Because instead of using these tools to look at one isolated protein in a test tube, we are using high-throughput data to map the entire massive interacting network of a living cell.
The whole web.
Right. But I have to ask, if you are looking at a chaotic web of thousands of interacting proteins, doesn't zooming out to the whole network make it infinitely harder to find a single target for a drug?
It might seem counterintuitive, I know, but zooming out is actually what prevents catastrophic failures in drug development.
Really?
Yes. If you only look at one isolated protein and design a drug to block it, you might administer it to a patient only to find the disease continues to progress anyway.
Because the cell found a workaround.
Exactly. The cell had 5 redundant backup pathways to bypass the protein you just blocked. Systems biology allows us to map the entire web to find the true bottlenecks.
The intersection points.
Right. The single critical intersection points in a disease pathway that the cell absolutely cannot bypass.
So what does this all mean? We've reached the final step. From bench to bedside, all of this molecular manipulation, proteomics, and pathway mapping culminates in clinical diagnostics and drug discovery.
And we see that immediately in diagnostic enzymology. When specific tissues undergo necrosis, they rupture.
And leak their contents.
Right, they leak their intracellular enzymes into the blood serum. By measuring the activity of those specific enzymes, we diagnose the exact location of the damage. For instance, if we see elevated levels of ALT and AST, we know there is specific hepatic damage, like hepatitis.
We're back to our patient in the emergency room at the beginning. You run an immunoassay looking for creatine kinase MB or CKMB. It's an isoenzyme specific to myocardial tissue.
So if it's spiking in the serum.
It confirms a heart attack.
Yep.
And the clinician's job isn't even done there, because tracking the decline of those enzymes back into the normal reference range is how you monitor the patient's actual recovery.
We also rely on molecular markers to monitor progressive conditions, like chronic kidney disease. CKD is managed in distinct stages, determined by the estimated glomerular filtration rate, or eGFR.
Right.
Stage 1 is relatively normal, with an eGFR greater than 90. But as nephrons fail, the rate drops. Stage 5 is defined by an eGFR of less than 15, indicating total kidney failure requiring dialysis or a transplant.
And catching that molecular decline early buys us the time to deploy therapeutics. The text walks through some brilliant case studies of drug discovery. Take hypertension.
A huge issue.
Yeah. The molecular target is often phosphodiesterase, or PDE, enzymes located in vascular smooth muscle. If you inhibit PDE, the muscle relaxes, expanding the vessels and dropping blood pressure.
And fun fact about that.
Oh, sildenafil.
Yes, sildenafil was originally developed to inhibit PDE5 for exactly this reason. But because it specifically enhanced nitric oxide in penile tissue rather than systemic circulation, it was pivoted to become Viagra.
It is a profound lesson in tissue-specific targeting. And another critical case study is atherosclerosis. Systems biology mapped the exact progression here. Atherogenic lipoproteins penetrate the arterial intima, where they become oxidized.
Right, which sets off an immune response.
Exactly. Macrophages consume these oxidized lipids and transform into lipid-loaded foam cells. And these foam cells accumulate to form the dangerous necrotic plaques that ultimately cause strokes. By understanding every single step of that pathway, pharmacologists have multiple distinct bottlenecks they can target.
And treating an infection requires a completely different mindset.
Oh, definitely.
because you aren't trying to fix a human biological pathway. You are trying to selectively poison a pathogen. The drugs must target the unique proteins of the bacteria to halt its replication without causing any collateral damage to the host's cellular machinery.
It really is an unbroken chain. Like, we started by calculating a standard deviation on a benchtop to ensure our data wasn't noise.
Yep.
We froze cells in milliseconds to avoid visual artifacts. We amplified DNA with thermal cycling and fired gold pellets into tissues. We mapped atomic structures using lasers and X-rays, all to find a single cellular bottleneck so we could develop a drug to save a patient from stage 5 kidney failure.
It is an unparalleled scientific triumph. But it leaves us with a really profound prospect regarding the future of personalized medicine.
Oh, how so?
Well, currently, clinicians rely heavily on population-based reference ranges. We are comparing a patient's labs against the average human, but as systems biology and high-throughput nucleotide sequencing continue to evolve, we are facing a massive paradigm shift.
Because the averages won't matter as much.
Exactly. How soon will clinical biochemistry stop relying on general population averages and instead diagnose you by comparing your real-time lab results against your own uniquely mapped, genetically predicted baseline?
That is the ultimate goal. Knowing mathematically what is normal for you and detecting the exact moment you deviate from it. All right, well, before we sign off, I have a mandatory review exercise for the medical learners out there. I'm going to fire off 3 clinical questions based on today's deep dive to lock this into your memory. Think fast.
Let's hear them.
Question 1. What specific mathematical equation is fundamentally required for preparing a buffer solution to stabilize the pH of a biomolecule?
We talked about this right at the start.
That would be the Henderson-Hasselbalch equation. Okay, question 2. A patient arrives in the ER with severe chest pain. Which specific isoenzyme immunoassay would you order to definitively confirm a myocardial infarction?
That tiny invisible enzyme.
You're looking for CKMB. And finally, question 3. In magnetic resonance imaging, what specific molecule's distribution, which makes up roughly 55 to 90% of soft tissue, is actually providing the physical image?
The most abundant one.
It is water, specifically detected through the 1H resonance. How did you do? I highly encourage you to review your notes and mentally apply these mechanistic concepts to your next clinical case or lab rotation. Because the deeper you understand the invisible molecular mechanics, the better equipped you are to change the physical outcome for your patients. Thanks for taking the deep dive with us.